In our final lecture for Chapter 1, we're going to be talking about the scientific method. So the scientific method is a way to understand and explain the natural world. So it's a method or a set of steps that scientists use to observe and then explain their observations in an attempt to answer questions. So the scientific method often starts with an observation or an inquiry. So someone notices something that interests them or asks why something happens, and that can lead to the scientific method. The scientific method can be considered sort of a general method where you informally test things and make observations and draw conclusions, or it can be a very well-defined set of steps, and often it is that well-defined set of steps that leads us to very specific data to answer very specific questions. So inquiry is just asking questions, looking for information, looking for explanations. Observations are things that you notice about the world around you. They're information that can be used to explain some of those questions you have. This can be a cycle. One can lead to the other. One can lead to more observations. Observations can lead to more questions. And so there's no set or defined way to start. But the scientific method starts with some sort of observation and inquiry. There are sort of two broad types of the scientific method, or two broad processes in the scientific method. One is called discovery science. So discovery science is simply studying the natural world, looking for patterns that lead to explanations of how things work, and it's often the first step in learning. So this process, this discovery science process, was really popular in the 1800s. There were many scientists who were traveling the world, looking around, observing things, describing what they saw. Some famous individuals are Charles Darwin, John Muir, James Audubon, and then even more currently, there are still people that are doing discovery science. Some famous ones from kind of mid-century and more current are Rachel Carson, Jane Goodall, and there are a lot of experiments and projects where people are doing their observation, performing different tests and things simply to discover information. They're not necessarily answering questions, they're just learning about the world around them. Another good example of this is the Human Genome Project. This was a project that started in the late 90s, early 2000s, and the point of this project was to sequence the human genome. We weren't really asking questions about it, we just wanted to know what it was made of, what was the pattern, how big was it, how different it was. So this is absolutely still a relevant process in the scientific method and in biology in particular. But we're mostly going to focus on hypothesis-based science. So there's a really good chance that you did this in elementary school, probably again in junior high, probably again in high school science, and this may be nothing new to you, and that's okay. Hopefully you'll get a better understanding of the process, and if this process is new to you, there are quite a few terms and things, and it can seem a little bit daunting because it is presented as a very formal, rigid process, but it doesn't have to be. With hypothesis-based science, you're attempting to answer specific questions with an experiment. So you want to ask a question and then design an experiment that answers that question that you're asking. And this sounds really easy, but it's often really hard to figure out what type of data you need to gather to answer your very specific question. But there is a process, a formal process that helps you work through this. And when we get to Lab 2, we will go through this process in formal detail. We'll do a very simple experiment to outline this process. Our book goes into a very specific case study to sort of outline the scientific method. And in this case study, researchers looked at coat coloration patterns in mouse populations in Florida. And I'm going to go through this in more detail after we go through the whole process, the background of the process. But I want you to keep this in mind, maybe even pause and read through it, skim through it real quickly just to kind of get an idea about this experiment, because I will refer to it as I'm talking in general. So just as a basis, researchers noticed that coloration in mice in the coast of Florida, specifically the Panhandle, varied based on the environment that they lived in. So you can see on the top there we have white mice living in a sandy environment and brown mice living in the inland soil and grassy environment. So they wanted to know what accounts for this variation, and one possibility is that it provides some sort of protection or camouflage from the predators. So we'll come back to this in more detail. So the basis for the scientific method is forming and testing hypotheses. So a hypothesis is not an educated guess. Often that's the way it's presented, especially at lower levels, and it's not a bad explanation, but it's not a guess. It is an explanation that you come up with to answer your question based on observations or background knowledge that you already have on the subject. So it is not a wild guess. It is a very specific answer based on what you already know. So this may require the researcher to go do some more research. It may require them to look at other people's studies. It may require them to think about or even do similar experiments before they can make their formal hypothesis. There are a few things, though, that are required of a hypothesis. And the most important thing is that it must be testable. So if you cannot test your hypothesis, it's not a good experiment. You have to be able to perform or design an experiment that specifically answers your question. And if you can't do that, you need to change your experiment or you need to change your hypothesis, because your hypothesis has to be testable. Another important aspect of hypotheses is that they're never proven. They are supported. So you could say, my data strongly supports this hypothesis, but a hypothesis is not proven. And that's because you simply cannot test every situation in which that hypothesis applies. Probably. Maybe you can, but very likely you cannot. And you also cannot test every future situation to which that hypothesis applies. So a hypothesis supports but not proves. This is really important, because if you say to me in your conclusions in your lab write-ups that your hypothesis proves something, I'm going to take off points. So make note of that. Some questions can't be answered, and therefore you can't make a hypothesis about it. So some examples of these are faith-based questions. You cannot test a faith-based hypothesis using scientific experiments. Some people will try, but the whole point of faith is that you have this faith without, or a faith in something without being able to prove it or to support it with scientific information. That does not mean that there is not evidence for a particular faith type question, but when using this formal scientific method, using a scientific hypothesis and in a scientific experiment, there are simply some questions that cannot be answered. Along with that, things like supernatural, I don't know, not necessarily abilities, but supernatural events is what I'm trying to say. Religious matters, things like that. Those don't fall under scientific method, and therefore a scientific hypothesis is not going to be testable. In a practical sense, when we're using our hypothesis in the scientific method, we are going to be using something called an if-then statement. So we'll talk about if-then statements a little bit more later, but an if-then statement is your hypothesis followed by a prediction of the specific results. So please take note when we go through the example here, and then again in lab, there's a specific way that you write your if-then statements. And you're going to use those to help you draw your conclusions and work through your experiment. As part of the scientific method, you are going to be gathering and analyzing data. Data is simply any set of recorded observations. So there are two major types. There are qualitative observations, and those are observations that describe what is being observed. So generally there are no numbers involved. It's more of a descriptive, you can think of it as descriptive words about what's going on. And then second are quantitative. So think of the word quantity. Quantities involve numbers. So these are going to be numerical measurements, quantities, frequencies, durations, total numbers, things like that. If we look at that example on the right there, that is Jane Goodall taking pictures of some chimpanzees, and then there's a picture of her journal there. So for qualitative observations, Jane Goodall might have written down something about what these chimpanzees are doing. They are probably picking fleas off of one another, or the mom might be holding or feeding that baby, and so she would describe that interaction going on there. But if she wanted to make quantitative measurements, she might measure how often these chimpanzees, these two chimpanzees, or the whole population engage in this activity, or for how long they do it, or maybe the number of insects that are removed from the other, or something like that. So both types of data are equally relevant to the scientific method, and the one that you're going to use or the one that you're going to analyze just depends on your experiment. For the most part in this class, we will be using quantitative. And then once you get your data, you're going to analyze it. You're going to determine whether or not you have supported or not supported your hypothesis. Very often researchers use statistics to help them determine if their data is significant. So you might look at two numbers and not really know if they are different enough to indicate that there's a difference in these behaviors in these animals, or if it's just simple chance. And that's what statistics does for us. We won't do too much statistics in this class. We do a little tiny bit when we get to our genetics section, and that's just to help us analyze our genetic events. But statistics is very often used in scientific process because it tells researchers whether or not what they're seeing is actually significant differences. So here's the big one. There are two major parts to the scientific method. One is forming that hypothesis based on your specific question. The other is actually doing an experiment. And a few minutes ago, I said that you needed to perform an experiment that specifically answers your question. And your hypothesis specifically answers your question. And they are related. And your data should be a direct reflection on the question that you're trying to answer. Now, one thing that tends to get students is they think that they're always going to prove, oh, see, there, I did it. They're always going to support their hypothesis. And that's not the case. Sometimes our experiments won't work in here. They're very carefully designed, and you might, quote unquote, know you have the right hypothesis, and then your experiment doesn't work. And that's okay. When we are going through the scientific process in this class and you guys are analyzing your results, I am not so worried about whether you have the correct hypothesis and whether or not you support your hypothesis. What I am most interested in is, did you choose or create a hypothesis based on your knowledge and understanding of the topic? And that means correct understanding and knowledge of the topic. And then, did you analyze your results correctly? So are you interpreting your results and determining whether or not you have supported your hypothesis that you made. There is one exception to that statement on whether I expect the correct hypothesis every time. There is one lab where your correct hypothesis absolutely depends on whether or not you understand the topic, and will warn you about that. But for the most part, I want you to be interpreting it correctly, using your data to correctly interpret whether or not you have supported your hypothesis. So this experiment part is the really important part. Most of the experiments that we will do in here are specifically designed for the class. They're set up because they work well. They've been doing them for many years, and so we sort of know what works and what doesn't. But often you have to start from scratch. You ask a question, you form a hypothesis, and then you have to design an experiment. And as part of designing that experiment, there are a few things you have to think about. These 5 points are really important, and students often mix them up and interchange them, so make sure that you know these 5 points. The first part of the experiment are the control and treatment groups. So the control group is the group that is sort of your standard or your comparison. It's usually the group that receives no additional treatment, or they receive sort of the normal treatment. So if you think about that mouse experiment, we've got these 2 different mice, and we want to know why their color changes. That white mouse in the white environment would be the normal situation, the one where that mouse is supposed to be. That would be your control group. And similarly, in the dark environment, that brown mouse would be your control group. Your treatment group are the group of individuals that receive a specific treatment. They're the ones that some sort of change has been made to that group, and you make that change in order to answer your question, to see the difference from that control group. So in this case, with these colored mice, your treatment group would be the mouse that's the different color. If you take that brown mouse and put it into the white environment, that's your treatment group. Your control is the white mice in the white environment. Your treatment group is the brown mouse. You've changed that brown color, or you've changed that fur color. You may have noticed that these are overlapping terms. Also important to the experiment are the variables. What are you changing? What is different between that treatment group and that control group? So your independent variable is the variable or the factor that the researcher changes as part of the study. And this is going to be related to your hypothesis. The dependent variable is the data that you get. It is the difference, usually, or the change that you see when you look at the control group versus the treatment group. And we'll talk about this more specifically in just a minute. Finally, this last one, we have experimental controls. So this is often mixed up with experimental variable, which is another word for the independent variable. It's also mixed up with control group. Experimental controls are also called confounding variables or controlled variables. So I'm sure you can see some of the confusion here. The experimental controls are the conditions or the variables that are not being studied that could affect the results. So they're called confounding variables sometimes because they are variables that could very likely affect the outcome of your experiment. And they are usually variables that you're not specifically testing for. And we'll talk about this with mice in just a minute. But part of good experimental design is reducing those confounding variables. That is, introducing these controlled situations into the experiment so that you can sort of limit what sort of other outside influences are going on. So as a really quick example, let's say that you're on a diet and you decide, I'm going to cut out carbs for a week and see what happens. So there's your independent variable, the amount of carbohydrates you consume, and then your dependent variable is going to be your amount of weight loss. So let's say that when you cut out your carbs, you also start eating low fat. Now you've introduced a confounding variable or an experimental variable, something else that could affect the results. So now you don't know if any weight loss is caused because of the reduction in carbs or the introduction of reduced fat foods. So you want to, in this case, keep everything the same except reduce your carbs, and that reduces those experimental, reduces the confounding variables. So by controlling how you do your experiment, you limit other outside influences. Finally, you take all of your data and you analyze it and you determine whether or not you have supported your hypothesis. Most of you are probably familiar with drawing conclusions, making conclusions, writing papers, and making a conclusion statement. For our lab specifically, there are a few things that you should talk about. And for every lab, when it asks you to make a conclusion about your data or did you support your hypothesis, there are several things that you need to address, whether or not it specifically states. So the first is, did you support your hypothesis? This is a yes or no question, and it should be stated as a complete sentence. Yes, I supported my hypothesis, but that's not enough. How or why not? So if you supported it, what specific data leads you to believe that you have supported it? Or if you have not, why not? Did something go wrong in your experiment? Is there a better hypothesis for this question? And are there any limitations in the experiment? So all of those things need to be addressed. And finally, from this, you also need to realize what you've learned from the experiment and whether or not you need any more further studies. Finally, we get to theories. So theories are broader than a hypothesis, and they are sort of these big, broad hypotheses that have been supported by a large body of evidence. And often people start to think of theories as true. There's something that have been supported so many times that you just accept it as part of everyday life and as truth. But really, theories are still a hypothesis. Remember, we said a hypothesis can't be proven. A theory is just a really big one that's been supported over and over and over again. But occasionally, some new evidence will come up that no longer supports that theory. And maybe that theory needs to be modified, maybe a new one needs to be made. And so the big take home here is that theories are big and broad. Think about the theory of relativity, the theory of gravity, theory of evolution. Those are all theories that have been supported by a wide body of evidence. I like this picture here. It says gravity, just a theory, and he seems to be floating in the air. I just find that funny, but gravity is actually a well-supported theory. All right, so let's get into this case study on the coat coloration in mice. So to start out, as I said, we have these white mice on the white sand, and we have these brown mice on the brown soil and grassland. So let's start off with the hypothesis that these researchers proposed. So their hypothesis was that coat color was well matched to the habitat that these mice were found in. And the reason for that is that coat color is an adaptation that camouflages them from predators. So this seems like a very likely explanation to me. If you can think about lots of examples of individuals who are camouflaged with their environment. So we have our hypothesis here that attempts to answer the question. And our question is not specifically stated here, but our question is, why does the coat color of these mice differ in this mouse population? So stemming from their hypothesis, we can make a prediction of the results that we expect. So if these mice differ in color because of predation, then we would expect that the mouse that doesn't match its environment would be more heavily preyed upon. So there we have some data. So our hypothesis attempts to give an answer or an explanation to our question, and then our prediction specifically gives us the data that we would expect if we are supporting our hypothesis. So, as part of experimental design, researchers built model mice. So these are plastic mice with fake fur, and put them in the natural environments of these individuals. And then they waited. They waited for hawks and things like that to attempt to attack or to prey upon these mice. So let's go through our important parts of our experiment. First, we have our control group. This is also called the experimental group. We have our control group, and then we have our treatment group, or our experimental group. So our control on the left in the white environment is the white mouse. You see that he blends in really well. He doesn't stand out nearly as much as that brown mouse. So that brown mouse in the white environment is going to be the experimental or the treatment group. So this is the one that's sort of not the normal. He's the one that doesn't belong in this environment. So think about what you expect of that mouse. And then on the right hand side we have the brown environment, and you can see that that brown mouse is very well camouflaged, and that white one sticks out like a sore thumb. So our independent variable here is fur color. So remember, this is the condition that researcher changes. So we are looking at fur color. We want to know why fur color changes or is different between these two populations. The dependent variable then is how often those individuals with the different fur color is preyed upon. Remember, our hypothesis says that this fur color matches the environment because it protects against predation. So we need to measure something about predation. So we look at our independent variable fur color, and then we measure predation on each type of fur color. In almost all of our labs that are specifically using hypotheses, and not all of them do, but many of the early ones do, we are going to be writing an if-then statement. So an if-then statement is a statement of our hypothesis followed by a prediction of the specific results. So one more time. Our hypothesis is that coat color well matched to habitat is an adaptation that protects mice from predators. So there's the answer to our question. Why are these mice different colors? The then half of the statement, so if hypothesis, then predict results. Our prediction would be then that mice who don't match their environment are preyed upon more frequently. So there's our data. We're looking at how often these individuals are preyed upon. So we are making a general hypothesis followed by specific expected results. Now, sometimes students have problem with this and they start making a circular argument. So this is the do not part of the if-then statement. So we do not want to say, if coat coloration well matched to their habitat is an adaptation that protects mice from predators, then mice will be protected from predators. We just restated our hypothesis. But when we introduce the data that we're talking about, we get a correct prediction of data. So if mice are protected, if coloration protects mice in their habitat, then mice will be preyed upon less frequently. Do you see the difference there? If not, make a note of that and please bring it up in class. So now we get to our results. So they left these mice for some number or some length of time in their habitat. And they looked at how often the mice that didn't match their habitat were preyed upon. And you can see that the mice that don't match their environment were preyed upon more heavily than the others. And this is where your statistics would come in, and you would look at whether or not this is a significant difference in predation, and very likely in the scientific journal that they published it in, there is statistics related to this. So I said I would go back to those experimental controls, your confounding variables. So there are other factors that could very well be impacting whether or not these mice are being preyed upon. That could be time of day. It could be maybe the shrubbery or lack of shrubbery around in their environment. So there are other factors that could be affecting this. And so let's look at time of day. So if these mice are out at a specific time of day, maybe they won't be preyed upon simply because their predator is not out at that time either. And so we would control for this by setting these mice out at the same time in each of these environments, and then determining how often they're preyed upon. And in that situation, then you limit the effects of time or some other confounding variable. So if we look at that data, we can see that the percentage of mice that were attacked is greater for the mice who don't match their environment. This supports their hypothesis that the matching coat color provides protection against predators. So we also see that those camouflage models have lower predation rates. So both bits of data are important here. It shows that by using the control group, you are showing that those predators will still attack those mice, but they are attacked at different rates. And that leaves you to support or not support the idea of fur color.